diseases which we are going to discuss is very popular. It's Friday evening. I have a pleasure to announce the award ceremony. Thank you very much. I also welcome all the participants of the 18th Assembly Healthy Moscow. Thank you for being here in Sklifosovsky Hall. I would like to recall that it's a large-scale event, medical event. We had more than 90 expert sessions and discussions in all the spheres of medical treatment. All people from 22 countries are speakers at our assembly. And before starting med talk from gene to disease, I would like to announce the award ceremony. We finalize the festival Formula of Life, which is held since 2012 among the medical workers, which allows experts to confirm their qualification to present to the wide audience the image of a contemporary doctor. Every person who dedicates his life to medicine is a hero, and today we would like to award the winner of the Specialist of the Year, doctor, pediatrician. And I would like to give the floor to Elena Petlyakina, Doctor of Medical Science, endocrinologist, head doctor of clinics of Moscow. Am I right? Applause, please. I would give you the floor for a welcoming word. Probably you would like to add something, and we would give a word to the winner. I'm thankful for such an honor to present an award. You know, I'm a pediatrician myself, then children's endocrinologist, and I believe that all the children's doctors can say that after graduation from this faculty, medical faculty, we all are pediatricians, and after that we are narrow specialists, we are specific specialists. And the person who won, it was a fair competition, and I believe that this is the best of the best. And it's time to announce the Pediatrician of the Year, Elena Gracheva. Congratulations. I believe that she's not here. Oh, it's not a problem. We will give a word to her colleagues. Our congratulations on that victory, the best of luck. And now it's time to start our session. Please take the prizes. We will give it to Elena. And I would like to give the floor to Elena. Med talk from gene to disease. Dear colleagues, allow me to start our med talk. We have innovative assembly this year, but it was difficult to imagine that at that table there will be head of Center for Molecular Genetics, Catherine Braunstein, and head of City Center of Orphan and Other Rare Diseases, Natalia Pechatnikova. And I will be a moderator. It's a great honor for me, and I would like to say that it proves that we use same protocols for medical treatment. We have same problems, and we are together with the leading world trends. Catherine, please, I give the floor to you.
Thank you so much for having me here today.
Dear colleagues, those of you who don't have headsets for simultaneous interpreting, please take the headsets.
I'm going to be speaking today about rare disease discovery. I have one disclosure where I do some consulting. I'm going to be about rare diseases at Boston Children's Hospital. There are around 7,000 rare diseases. You'll see different statistics for that kind of number, but in the general vicinity, of around 7,000, and 80% of them are genetic in origin, and 50% of those are pediatric in presentation, which means in the United States, around 30 million people live with a rare disease, and around 300 million people worldwide. So we can do much better in terms of disease diagnosis. On average, it takes 7 to 12 years to diagnose an undiagnosed patient. 8 specialty physicians, they go through 2 or 3 misdiagnoses. And we have huge problems where physicians don't have the time or the information to work with these complex cases. And it takes a major emotional toll on both patients and caregivers trying to figure out what's wrong. So at Boston Children's Hospital, we're around a 400-bed quaternary care magnet hospital where we do everything except for burns. They usually go to a different hospital for that. And we do a lot of research and have around 500,000 clinic visits per year. Patients come from all over the world to Boston Children's with 2 fundamental questions usually. What is wrong with my child, and what's next? And a lot of times, genetic testing can be used to answer these kind of questions. We've developed a precision medicine service at Boston Children's, and this is something almost as simple as figuring it all out, all the logistics. So if you're coming from the Middle East or Russia to Boston Children's, you want to make sure that your medical records are there before you get there. If you had genome sequencing, that the sequence has gotten to Boston Children's and has been analyzed before you get there. If you have a multi-system disorder, that you are seeing all the specialists on the same day and not 6 weeks apart, requiring you to fly back and forth again. Now, all of these sounds like very common sense kind of things to do, but they're surprisingly quite difficult when you're dealing with a complex hospital seeing thousands of patients a day. So, and especially when you're dealing with all these different languages. So we've created this new service where we have people dedicated to handling these kind of cases and the logistics behind them. But sometimes even that fails. You come and you're seeing all the specialists, you have your genome sequence, you've seen everyone at Children's, but they still can't figure it out. So there, we're lucky in that we have a streamlined transition to the research realm. And I am the scientific director of the Manton Center for Orphan Disease Research at Boston Children's, where it was created when Pankaj Agarwal, my colleague, was realizing in the NICU, he is a neonatologist, patients were being born with seemingly genetic disorders, but there wasn't really any way to get them into research programs. Either the burden was on the clinician to write an IRB for that patient, which takes a lot of work and takes a lot of time, or the patients were being crammed into established research programs where they weren't necessarily the perfect fit. So Alan Beggs was working with the Manton family where they wanted to give a donation to start a center. And here we were able to have a virtual center where you have a blanket protocol where any patient with a rare and orphan disease can enroll, and they don't have to start from scratch to create a patient IRB. So the Manton Center is a philanthropically funded virtual center that is over 10 years old now, and is focusing on understudied diagnoses, and we've recently expanded out so now we can take common diagnoses that have influence on rare diagnoses. So for example, I myself work on very early onset psychosis in children, but now I can enroll someone with a normal onset schizophrenia within the same family and look at them in context to their sibling that has very early onset. And we have a senior scientist program, endowed chair, innovation funds, pilot project awards, which I think is very important because to keep funds for innovation flowing within the hospital keeps things very innovative. Fellowships, a visiting science program, and the Gene Discovery Core Protocol, which is what I want to speak about now. Again, we have broad inclusion criteria. Anything with rare and orphan disease can enroll, and it's really designed to enhance the clinician-researcher relationship. You're allowed to do virtually any kind of molecular and functional testing tailored to the case. And the brilliant thing here is that samples are able to remain identifiable, or patients can remain identifiable, which is key for moving things from the clinic to research. And then if we figure something out, we can move it back to the clinic as quickly as possible, and we don't have to go through the re-identification process. We have infrastructure to return results to families and into the medical record. We have genetic counselors on staff. And then we also have staff to support project development and to oversee the enrollment process and to maintain the protocol. And this is key when you're working with rare diseases, because sometimes you'll be working on a gene and then suddenly you're at a place like this and you meet someone who's working on the same gene as you, and they have 5 patients and you have 3 patients. And being able to collaborate and have the patients enrolled in the same program and being able to work together is paramount to the success of the program. So we have over 5,000 participants enrolled from over 2,000 families. We have referrals from pretty much every department in the hospital, as well as, we actually just got the last referral. It was New Mexico, so from all 50 US states and 6 continents. And importantly, less than 7% of families approached have declined participation. And really, this goes to show the severity and the urgency of this research, that patients really are quite willing to donate of their time, their blood, whatever we need in order to work with us and hopefully find answers. And we've had results returned to over 200 families. I thought this number was actually quite low. And this is result return, when I clarify with the genetic counselors, is in the classic sense where they were able to give the gene name, the mutation, and have it in the medical record. We have many, many more cases where we have a candidate. And there we're not running around telling people candidates because it can give the false sense of what's going on. So this number seems a little misleading. It's because many more are in progress and not quite there yet. The benefits are that the results can be returned. You don't need to travel to Boston if you have cases that you want sequenced or to have team investigate. The patients can be enrolled over the phone. We have a language line. And it can be international, and there's no cost to patients, which also helps when working with a lot of cases where they're in countries where access to genetic testing is a problem. I spoke about this earlier today, quickly. This picture of Mila is shared with the family's permission. This is a beautiful child who unfortunately has started around age 7 and a huge, or actually 3.5, a huge loss of motor skills, vision loss, seizures, cognitive decline. And it looked like Batten disease, but they could only find one mutation when it is a recessive disorder, and you need 2 mutations in the CL7 gene in order to manifest the condition. So after going to several different doctors, the family ended up at Boston Children's Hospital with my colleague, Tim Yu, who looked at the sequencing and was able to identify in the raw reads, not looking at the BAMs. We went back to the original raw data and saw what looked like a retrotransposon was interrupting the gene and causing a premature stop codon. And that was that missing second mutation that they couldn't find through normal sequencing. So he was able to design an antisense oligo and hopefully enable exon skipping. So and restore normal gene splicing, skipping over the problematic area. This was a crazy timeline from the mutation identified to the first dose. They were able to go to Washington, because this is a US-centered program, and get the necessary permissions for this experimental treatment. One thing they did say is like, can you do 4 months of mouse toxicology? And he's like, no, we don't have 4 months. The patient's going to pass away. And so they were able to get the permissions. And from proof of concept to the first dose of this tailored therapy was 4 months. So this slide at first glance doesn't seem all that impressive, that there are patients still having seizures. But then when you look at the details, it is actually quite striking, that the seizures went from very intense, very long in duration, to have much less intensity, less frequency, and she actually did have a couple seizure-free days. And it continues to work. Unfortunately, it's never going to reverse the condition, but the parents are so happy that they've been able to stabilize the disorder, and Mila, even though she still has Batten, has this custom therapy just for her mutation. And Tim was able to publish this in New England Journal of Medicine a couple weeks ago. I myself work on childhood onset psychosis. One of my interesting cases is ATP1A3, where the proband was a 9-year-old Caucasian boy with a history of selective mutism and severe aggression. He, at 2 months of age, was in physical therapy for decreased muscle tones, but motor and speech milestones were within the normal limits. At age 2, he exhibited severe head banging and then seemed to have pervasive developmental disorder and depression. Around 6 years of age, he fit the DSM-5 criteria for childhood onset schizophrenia. He had lack of emotional control, separation anxiety, and mood swings. And I leave these descriptions here not to shock you, but to show that this is a truly psychotic child that is not on the realm of, like, perhaps it's abuse, perhaps it's aggressive imaginary friend, something that's going on. But there is some gray area for some cases, but this is a true case of psychosis and behavioral regression. So the patient was referred to the developmental neuropsychiatry program, where I work with a physician named Joseph Gonzalez-Hedrick, and we did whole exome sequencing and identified a de novo in the gene ATP1A3. Now, around over 5 patients have been identified with de novos presenting with very early onset psychosis or severe behavioral disorders. But it wasn't a slam dunk when you look at this and, oh yes, this must be it, because ATP1A3 is tied to several disorders, and when you look at the case studies, there are behavioral effects that are mentioned, but usually it's not the first thing mentioned for mutations with this disorder. It's linked with rapid onset dystonia of Parkinsonism, Kaposi syndrome, and then also alternating hemiplegia in childhood. But the patient that we saw had none of these things. And when you read the case studies of alternating hemiplegia in childhood, the patients are impaired. They do have behavioral difficulties, but usually behavioral difficulties are not the first presenting symptom. So we've gone on and gotten lymphoblastic cell lines established, turned them into iPSCs, into induced neurons, cortical neurons, and we sequence them. When you run it through just the off-the-shelf David sequencing platform analysis tool, it shows that schizophrenia is one of the top signatures for the gene expressions, which was nice to see. It kind of showed that, yes, our cell lines seem to be working quite nicely. And then we were able to CRISPR correct the mutation in the patient, and we're now in the midst of phenotyping the CRISPR-corrected lines compared to the mutation lines and comparing them to the unaffected father. If we are able to correct the phenotype seen in a dish, we are going to CRISPR in the mutation into the father's cell line and see, again, if we can induce the cellular phenotypes and be able to quantify them well. And this work is being done by Rich Smith, who was a postdoc in Christopher Walsh's lab and now is promoted to instructor and is starting a lab of his own. My overall message here is, if we aren't keeping up with this kind of technology, and our patients know about CRISPR, they know what technologies are out there, and that really it's a question of access and time and getting their case to the front of the line. And if we don't keep up, their patients are going to go it alone and find companies that are willing to work with them. And here's one example of a precision medicine platform where they take the patient sequencing, identify identify targets. They're doing pharmacogenomics and disease screening in a dish, especially for something like uncontrolled seizures. They're able to do high throughput screening and see the best medicine for the patients. Here, we're actually partnering with this company on ATP1A3 and are going to do enhanced phenotyping and enhanced phenotyping of the CRISPR-corrected cells. And because, again, we're looking at a channel, being able to see if we can alter, or actually we're looking at a pump, we can see if we can alter pump function. Here's another exciting case that my colleague Alan Beggs is working on, where he's devoted his life to X-linked myotubular myopathy. And this is a disorder that's in about 1 in 50 newborn males. It can have a range of severity. Some 50% have a mortality by 18 months. But then if they are one of the milder types, they can survive up 10 years and beyond. Motor milestones are delayed or not achieved. They can't sit without support. A lot require breathing support and have a high rate of healthcare utilization, hospitalization. So here I'm going to botch this if I try to go into too much detail, but really Alan was able to design a virus vector that stably expressed the protein and under control of the Desmond promoter, and was able to first see in dogs. I don't know if you can play the movie. I don't have ability to press play here. I don't know if you're able to see it, but we have a myotubular myopathy dog versus a normal, and it's probably not going to work, but the difference is quite striking between this dog that looks completely impaired and one that looks normal. But under the control of the promoter, they are able to give this therapy, viral therapy, to the dog, and the dog looks pretty much normal. In a heartwarming story, some of these dogs have been adopted by families with children with the disorder. So now you have the child that's using the treatment and having with the dog that has the treatment. So Alan's been working with Audentes Therapeutics that has been doing this type of gene therapy for several diseases, though X-linked myotubular myopathy is the best, furthest along. So here are some of the criteria for enrollment in the study, and I want to go quickly just to show, this is my favorite slide, where it shows the progression of each patient. And some of them are able to progress to walking. Some use assisted support in order to walk. But when you talk to the patients, every single one is thrilled beyond belief. And if you ask them what is the most exciting thing, change, they say they're able to scratch and itch. And that just kind of made me tear up talking to a patient. And when it goes to show the importance of actually talking to patients and understanding what's important to them. Like Alan is such a perfectionist that he sees anything less than being able to have non-supported ambulatory change be a success story. But here, patients to be able to scratch their face when something is itching is a huge milestone and incredibly wonderful to them. So Boston Children's is quite innovative in that they do not balk at us working with pharma companies, with for-profit companies, as long as our missions are aligned. And I just completed a really exciting, I think, 3-year program working with Amgen where we were screening patients looking for targets in pain. In the United States, we have a huge problem with opioid abuse and the lack of good pain treatments. So we were finding patients, especially children, with chronic pain and insensitivity to pain, and doing whole genome sequencing and looking for new pain targets. I worked with my colleagues that were skilled mouse technicians, and they would make mouse models. We would test the mice and see if we could come up with any targets for new treatments. Here's one case where the child would repeatedly get frostbite because he didn't feel pain, required no analgesic for a tonsillectomy, enjoys exercise, but when he gets hot and he becomes dizzy and tired, he has to wear cooling vests, doesn't like the feeling of showers, And he's very high intelligence, and which actually made him kind of difficult to phenotype because he knew when he should be feeling pain. So when they would do some of the tests, he would score as being normal, even though we knew he wasn't, because he knew that, oh, this should hurt. I shouldn't be doing this, which is one of the benefits of working with someone that was kind of very brilliant. The disadvantages of working with someone like that. It made it a little more difficult. But when we went through we were working with Decode with this in Iceland, and Iceland's extremely unique that a large population of the country has been sequenced, and they have very complete medical records. So when we had a gene candidate, we were able to go to Iceland and query their database to see if they had a same gene candidate. They had something that wasn't identical but quite similar, and we were able to contact the family, and we're in the middle of phenotyping them for the same, see if they have the same pain disorder that our child first does. We're also working with Alexion Pharmaceuticals, going from HPO, which is human phenotype ontology, to diagnosis. And Alexion has an interest in being able to shorten the length of the undiagnosed journey for patients and be able to perhaps identify them just through the medical record and reduce the need for sequencing. So they provided some funding for Boston Children's to translate the medical records into HPO. We were extremely excited about this because we can build our own algorithms and have kind of ontologies to understand the relationships between patients. And Alexion was thrilled because they were able to receive de-identified data to assess the performance of their suite of algorithms. And it's one of those situations where it's a total win-win, and due to the open-mindedness of the hospital, we're able to work with this pharmaceutical company. Human phenotype ontology is really exciting for us because we're able to work with it to even go directly to the patient and get even more rich phenotyping. I've worked with the Monarch Institute and Melissa Handel at OSU to develop plain language symptoms for computable phenotypes. So everyone in this room probably knows the word strabismus, but the average patient doesn't know that. They know lazy eye or cross-eyed or something's wrong with my eye. So being able to take that information and code it into the parent term as something that you can go back and ask the patient more about, but they're able to fill out in the comfort of their own home is really an exciting thing, especially when we're talking about doing deep phenotyping that can be done remotely and not necessarily in the clinic. We're never saying that patient-centered phenotyping is going to replace the clinic visit, but it can augment the clinic visit. And especially at a tertiary hospital like what we have at Boston Children's, and I'm sure in Moscow too, that clinicians no longer have the hour-long visit to sit and discuss with their patient everything that's bothering them. So if they can at home fill out everything, and then have these HPO terms for follow-up questions, or a sheet with HPO terms that they can take to their clinic, their local clinic. It may provide better resolution and hopefully increase diagnoses. So I've worked actually with John Brownstein, full disclosure, he is my husband, Chief Innovation Officer at Boston Children's, to develop Phenotyper, which is patient-provided phenotyping. Patients can fill this out at home on their own, and it codes it into the back end. Interestingly, we started with just the layperson phenotypes, like putting lazy eye, not strabismus, and we had to go back and add in the medical terms because the patients get so educated along the way of these diagnostic odysseys that they were getting frustrated that they knew the medical terms for what they had and we didn't have them listed. We've tested this with the 16p 13.11 copy number variation registry. This is an interesting copy number variant in that it can have a huge distribution of phenotypes where it's associated with schizophrenia, developmental delay, microcephaly, macrocephaly, epilepsy, or you can be completely fine. So, and also looking on the Facebook chat groups for 16p 13.11, the patients were discussing phenotypes that weren't in the medical literature. So we wanted to quantify that some way. And the way to do something like that is to go directly to the moms. Our patient organizations get things done. The mothers were able to rally everyone to enroll in Manton Center for Orphan Disease Research, to fill out Phenotyper, and we've already seen that GI issues are a major issue with 16p 13.11. Also late age bedwetting, which are things that that are not currently in the medical literature, but only when you went directly to patients and try to quantify what they're experiencing, we're able to figure that out. So, and again, this goes to show that rough phenotyping as enhancing the route to diagnosis is something that shouldn't be glossed over. So, for example, we have implications of diagnosis on a care plan. Insomnia is part of schizophrenia. It often goes undiagnosed because frankly, there are more pressing things to worry about when you have a schizophrenic child. Decreased time in REM actually has a huge impact on behavior. Getting the correct antipsychotic can actually perhaps affect REM too. But a lot of times parents don't know if their children are sleeping properly. So we had a pilot with Withings, now Nokia, where patients were able to get a pad underneath their bed and see if they are sleeping properly. Now, we don't think this in any way replaces a sleep study where you're sleeping in the clinic, they have you hooked up, they know exactly what you're doing, but you're able to figure out a good night versus a bad night. And in 2 of our patients, we realized that they weren't even getting 6 hours of sleep a night on average. And when they're an 8-year-old child, they need much, much more than that. And the Withings Aura was able to figure out that they were getting up in the middle of the night, walking around, sleeping on the floor, things that could be addressed, and having better sleep means better behavior. So in conclusion, we think that investing in architecture, especially when you're dealing with N of 1s, with rare diseases, is paramount. You want to take the serendipity out of matching to partners to overcome bottlenecks. When I was doing my PhD, I worked on one family for 6 years. Now I have 2,000 families. So figuring out how to route patients, how to get the patients to the clinician, to the researcher, to the pharmaceutical company, to the therapeutic company that is going to be able to address their concerns and make their lives better is paramount here. Collaboration is the key. I'm hoping to meet as many people as possible here because having an N of 1 and being able to turn it into an N of 10 is an amazing thing. It means so much in terms of diagnosis and treatment. And next-gen sequencing is only part of the story. It's not enough to just diagnose a patient. Like, I've recently worked with 3 other institutions and found a new gene for what we think is a behavioral disorder in a few families. And we're like, and the overwhelming thing is, so what? So now are you going to treat this? How are you going to do this? And it's kind of upsetting to hear, but it's a really important thing to hear, that it's not, the story doesn't stop because you figured out what's going on. The story has to now go to custom therapies and something that's going to make things better. And I'm not just meaning pharmaceuticals. With ATP1A3, actually the most important thing, which we found out, is that the child is able to take nap breaks throughout the day. And somehow having a short nap resets the brain, and his behavior is much, much better. So it's not all pharmaceuticals. And then being able to stratify patients for clinical trials, to be able to get deep phenotyping, accurate phenotyping, and be able to truly understand what's bothering the patient that way. And then relationships with industry are valuable for all parties, and I'm lucky that I'm at an institution that understands that. So right now we have care, we have discovery at Boston Children's Hospital, and hopefully we're going to move them into one and the same thing. And I think that's why we're all here today, is that we want the same thing for Moscow. Thank you.
Thank you so much. It was a really interesting, informative report, and even broader than we expected. Because we referred the issue of children, and a lot has done. Psychosis, child psychosis. We have not only neonatal but selective screening on inherited diseases, and we used to look a lot hereditary diseases, and there were lectures here. Everything is done for the patient to find his proper doctor and to have proper screening, proper treatment, time of targets of therapies, expensive therapies, the risks, the benefits. These are the questions. Now let's ask questions to Catherine before we switch to Russian experience. Colleagues, I do not see everyone. If you have any questions, do not hesitate. Any questions, please? Yeah, here we have one. Please introduce yourself. And mic to the speaker, please.
Не слышно вообще. Можно микрофон? Вообще не слышно.
Hello, my name is Irina, and I have two questions. Is there any link on the level of genes? Can you find the diabetes of children before they actually get this disease? And the second question, when both parents had are vaccinated, can we find out that some genes, that the child can have some genes not to vaccinate him? Catherine, would you like to ask and to answer? Yes.
Diabetes is a very interesting disorder. My colleague at Boston Children's, Jason Flanick, has devoted his life to common variants as they contribute to diabetes risk. I don't feel qualified to actually answer more than that there are different forms where it's very strong, runs in the family, looks like it almost could be a dominant disorder, but I'm hesitant to say any more than that. And then in terms of vaccination, I'm going to always say to vaccinate your children.
Вы знаете, можно я помогу?
If I may, I would add a bit to it. Diabetes is a polygenic disorder. Unfortunately, today, and what is polygenic? Yes, I'm sorry. That means it is coded by a combination of various genes. Combination of genes and the environment. Most orphan disorders are described by one gene, and gene-specific therapy is aimed at correcting the activity of this very gene, the pathological activity of this gene. Child diabetes, the first type, is autoimmune disease. Polygene is implemented there because it's autoimmune, the aggression of immune system against antigens of the body. That's why it seems to me that, and there are types of diabetes: monogene diabetes, neonatal diabetes, which is described by one gene and what is not connected with destruction of beta cells, of liberation of insulin. But that's an absolutely different diabetes, and it is manifested up to the age of 6 months, and it does not request insulin therapy. It starts with insulin therapy, then special research is done, and the patient is switched into the therapy, doesn't need some gene or expensive therapy. The patient needs a therapy which helps to free the insulin which is not there due to defect of a gene. That's why I don't know to which extent, but I can say that unfortunately I don't think the gene therapy can reach the child diabetes type 1. Monogenic therapy diabetes can be cured by drugs already. As for vaccination, vaccination is performed against infectious diseases. We can get into discussion that one and the same measles can give some aggravations to the brain and the other child can show no negative symptoms. But I think that worldwide infectious diseases were really a big problem before total vaccination in the 20th century. Europe was dying out due to infectious diseases. That's why the only way for prevention, I absolutely agree with Catherine, you need vaccination and reduction of the level of vaccination, which happened in Russia in the 1990s. It was reduced due to a number of reasons. As a result, we have what we have. The only way to prevent and to control, so to say, manageable infections, especially dangerous infections, is vaccination. The examples are numerous. A classical example is smallpox. We defeated polio and so on and so forth. So as far as I understand, if you already have immunity, it is not transferred with gene. No, they are not inherited. That's a Lamarck theory. No, it does not work this way. No questions from the audience?
There is a question. My name is Marat. Probably my question is not about orphan diseases, but I'm just curious what do you think? You're being classic genetics experts. What is a polygenic risk?
It's interesting to know your opinion about the polygenic risk scores.
So that's a fantastic question, and I think if you had asked it to me a couple months ago, I would have had a different answer. But I am now, with the movement to genome, all of a sudden we can do the PRS, the polygenic risk score, in addition to looking for the monogenic cause without having to send for an extra test doubling the cost. And we're adding this in. So for the first time, I'm looking at monogenic causes and copy number variations in my patients, and also doing the polygenic risk score, and hoping that that's going to have some informative, some new information about, especially the incomplete penetrance for some of these cases. When you have two siblings with the same copy number variant, one has the disorder, one doesn't. Perhaps polygenic risk has something to do with it. I'm hopeful that that's going to have some impact. I can't say it's something that I think I underestimated its importance beforehand. And I hope that's true, and I'm going to be able to go back and look at these unsolved cases and these incomplete penetrant cases and be able to explain some portion of them. So I'm not sure.
Thank you.
Other questions? Good afternoon. Thank you very much for such an interesting topic. I would like to somehow, you're a very beautiful woman, and you discuss some complicated things which are not clear to us actually. Let us talk about health. We have a man and a woman and a child, and the child has a gene mutation, and this child found a wonderful place where his disorder is analyzed. What do we need to do in addition at home with any mobile phone application? What shall we do to be healthy?
That's a very interesting question, especially because the home makes a huge difference. And with even behavioral disorders, having a supportive home, reducing stress, having the school be aware. When I was talking again about the naps, having a school that's okay with that, that can mean the difference between success and no success. So as for apps, I think some of the other sessions that we had earlier today were talking about, yes, there's this whole avalanche of apps out there. How do you separate the good from the bad? And unfortunately, I don't have a good answer for that. I know that the ones developed by hospitals and by reputable people, like, I have access to those, and I think some of them are quite wonderful. Children's Hospital now is like experimenting with sending people home with robots and being able to interface with their doctors, like even just for a minute, during the day, which would be incredible. Just counting number of seizures on their phone and being able to have an output and understand when, okay, we changed the medication and the seizure rate went up. That needs to be addressed. On the voice health, they talked about using Alexa to analyze voice, and when there's a change in the speech patterns or understanding, like, okay, they seem to be yelling a lot more, all of a sudden it's time for an appointment to come in. I'm very hopeful that there's going to be a lot of inventions and interventions that are going to make the home life easier and more successful for patients. But I think that's a huge issue.
Я, наверное, могу добавить.
I could probably add that there are now some general recommendations because everything depends on the particular disease and the very important problem which every family faces who has a patient with the hereditary disease. The key task is to live with this disease. It's a huge task, it's a huge challenge, which is a great impediment for many people. It's everyday job. When some diseases require some dietary nutrition and parents need to calculate the ratio of food, sometimes we need dietologists, the experts, those who know the patient. They shall be responsible for that, and they should give you answer. What are the weaknesses? What you should pay special attention to at school, at home? What should other family members pay attention to? Sometimes the whole family shall be on a diet in order to make it easier for the patient. If a patient, a small patient, a child is ill, we should be taught how to deal with it. I was a head doctor at School of Diabetes. We train how to calculate the bread unit, how to make insulin injection. I agree with Tatiana. As for the question about health, how not to get sick, I would say. This is the most important. This is a culture of wholesome nutrition, healthy lifestyle, personal gadgets, personal approaches of pediatricians to behavior of healthy children. It's not quite a topic of our session, but it's a separate area. It's a huge. We need to learn how. And the question to everybody: is strong spirit important? I don't know if our foreign guest will understand, because it's a very Russian saying. Your will and your spirit, how important it is in case of disease. I can only say that there are diseases, not diseases, but states, disorders, which are a condition for developing of many diseases. For example, weight. You shall control your weight. In order to lose weight, you need the will and you need a spirit strong. And can I ask Catherine what is spirit and the will for her?
I think I get it. I actually grew up in a Finnish town, and they used to call it sisu, I guess, or so spunk and spirit. We're actually measuring this in our pain population, and it's resilience to pain, and the ability to overcome pain, and for the parents to stay positive and work and be able to cope with this difficulty. And what are the factors? Because you can have a polygenic risk score and a predisposition towards mental illness, but somehow persevere and succeed. And what's separating those families from the ones that actually have quite a harder time with that? And if we can quantify it and figure it out and figure out how to apply those principles to everyone that we're seeing. Probably not a perfect translation, but it was the best I got.
Yeah.
Еще, если можно,
Can I answer questions? Number one is that you were talking about personalized drugs, personalized therapy based on the results of gene analysis. You are talking about rare diseases, about unique one-of-a-kind patients. Is there possibility to come to your hospital from other country, or you work only within the US?
All the entire world, and so, when you're working with rare diseases, you have to be global, because there might be one patient in the US and one in India and one in Moscow, and hopefully we can all get them together and look at 3 instead of 1.
то есть это возможно включить пациента.
It's possible to invite a patient, for example, to involve patients, for a patient from other country to take part in the.
Yeah, a lot of times they don't physically come. We send blood collection kits. We work with their physician. We do a Skype visit. We stay in touch that way. I would say most of our patients at this point aren't local. If they're not in the Boston area, I'd say a huge percentage are actually in other countries.
Спасибо. И еще вопрос.
Thank you very much. And another question is about the approach to diagnostics. Since you were saying about rare cases when you tried to NGS, and when you say NGS could help to answer the question, when a patient comes for the first time with complaints, with some problems, do you think it should be the complete exome, or you should start with some simple genetic tests, or the full exome is something that we should start with?
Such a great question. Okay. Before, I would say start with the gene candidate and just sequence that gene. But now it's almost cheaper to go straight to the exome. And I'm talking research. I'm not doing a clinical exome. The research exome, and then if you sign something to CLIA validate it, that's what our law is in order to do the result return. But the prices are dropping so rapidly that I see in the near future that a genome is going to be the first line, just because why not? And then you might throw out everything but the exome, so you're not dealing with a huge file to look at on the first pass, and then if you don't find it, then you go back and look at the genome. I still don't totally believe the copy number callers for genomes. I still want to do an array a lot of times, but hopefully they'll catch up quickly, and I'll feel comfortable with even doing copy numbers and everything from the genome.
Тогда, и если можно, несколько.
And some more questions about.
What do you think?
Я думаю,
I will have a presentation on the algorithm of diagnostics, and it's related with the fact that the prices do not go down as fast as we would like it to be. That is why we start with the simple tests, with the gene candidate, etc. And I'm dealing with the hereditary metabolic diseases. I should start therapy as soon as possible. And my question is, after results of the full exome, you have a huge amount of data about some diseases, about that we know that some diseases are not curable, they will develop in the later when with aging. do you think we shall tell patient everything what we do not have right to tell? Because after full exome, you learn a lot about a person. What shall we actually tell a patient and what we shall keep secretly? I don't know.
Практический вопрос.
Very practical and ethical question.
I think you have to know the patient's wishes. Some patients want to know everything, and they want to know if they have a SOD1 A4V mutation and they might have 18 months to live. And they want to know it because they want to quit their job and travel the world. Or if they have a BRCA mutation. We have a checkbox where you want to have that information or you do not want to have that information. I myself, when I did my own genome, that was the first thing I looked at. Do I need to quit my job? No? Okay. And then I went back to work. But I understand that not everyone thinks that way. So we actually do run into cases where we accidentally see something we aren't supposed to see, and then we have to go to the IRB because it is actionable. And even though they said they didn't want to know, what's the ethics there? Where, like, you know, what can you do if you can potentially save someone's life? But they said they didn't want result return. And I think those are complicated cases that I back away slowly from and let the experts deal with them.
Да, спасибо. У нас просто, к сожалению,
Unfortunately, our method of full exome is very widespread, but we don't have these documents. The patients, you don't inform us what they would like to know. We don't have these documents because I suspect that not everybody could guess, could predict what answers can the result, can the analysis give. And my last question. It's quite a trendy technology. It's expensive, full exome, but nevertheless it's very trendy and we have some understanding. If you do full exome, you will learn everything, your diagnosis, and you will live differently. You will have a new life. And some parents, some young couples, when they get married, they think that it's a right thing to do full exome for the both partners to learn which diseases they have to plan pregnancy. I don't know if there are such practices in other countries. My question is, if we find diseases, mutations, gene mutations in both, for both partners, but we cannot decide if this mutation dangerous or not. We cannot determine will it become a disease or not. Do you think there should be prenatal diagnostics in this situation?
So if you're talking like macular degeneration or something where, you know, it's not the most serious, or if they're talking like sickle cell anemia, or I guess it depends on the disorder. I would say it's also a very private decision between the couple, and hopefully with thoughtful intervention or discussion with their healthcare providers. The VUS, the variant of unknown significance, is a huge issue, and I'm sure we're working with metabolic disorders. It's life or death. Where in my area, we see after it's happened, so I don't deal with that kind of question a lot, and I think I'm more going to be interested in your thoughts and how you handle it, because it is such a huge issue. One thing in the last year, just through my personal life, I've had to deal with friends with who are going through fertility and working with IVF clinics, and I was kind of shocked at the lack of transparency. And they're saying, oh, you should use a donor egg. And it's like, why? And they're like, well, because they said you need a donor egg. And I think that needs to be hugely addressed. And like even just explaining the concept of a VUS to somebody and trusting them to understand it and make a thoughtful decision on their own, and not think it's too complicated for them to understand. But again, I'm not speaking as a professional here, and I'm really going to be interested in your talk.
Я
I'm not directly consulting families. I work in stationary clinic, and I face those. My patients are people to whom it has already happened. And when we recommend something, if we give some recommendations or we need an advice of our colleagues, a huge problem is considering it's invasive. Prenatal diagnostics, it's invasive, and the most spread problem is mitochondrial disease, type of hereditary disease, when we have no idea will this mutation lead to disease or not. Even after prenatal diagnostics, we cannot give a 100 guarantee to a family, but the family expects that, the family needs to hear that answer, and from this point of view it's very complicated. Dear colleagues, you have a very interesting conversation, you have a very interesting dialogue. I believe you will have an opportunity to continue it, because we all will be available. What we need more time for other questions. And I would like to give the floor to Natalia Pechatnikova, head of the City Center of Orphan and Other Rare Diseases. Dear colleagues, dear participants of the section, my presentation is more simplified, I'd say. We deal with diagnostic of hereditary metabolic diseases. I will tell about simple methods and techniques which could be applied for diagnosis of metabolic diseases. I will start with 3 cases. I will repeat them. It could seem like there are 3 similar stories, 3 similar diseases, but at the same time these diseases are different. 2 of them can be cured if diagnosis is at time, but unfortunately if we do not do that, it leads to death. And there is a symptom: hyperglycemia. Our section is together with the endocrinologist. 3 patients, same sex, same age, from different countries. All of them have problems with the liver and the hyperglycemia. The tests were same, but results were different. To our point of view, I would like to recall you how to make the correct diagnosis. We cannot do it without a laboratory, only on the level of clinical symptoms, and we can make a correct diagnosis. What tests do we have? There are levels of metabolic diseases. Something that Catherine was talking about today is about DNA and RNA. Next level is proteins, enzymes, and metabolites, which also could be measured. And advanced technologies allow to do that on the basis of a small number of biological material. Here you can see the evolution of genetic tests from 1 gene to multi-genes tests, and what we call target panels, and from them to exome and genome, from 1 enzyme to multi-enzymes test, and proteome, from 1 metabolite to metabolome. Today we are at this stage. Exome, proteome, metabolome is available from the point of view of technologies, but it's quite inaccessible from the point of view of practical healthcare due to high cost.
The easiest way which is used for diagnosis of hereditary diseases is the spectrometry, tandem mass spectrometry. It is done quite rapidly, and in fact The analysis takes a few minutes and does not require a lot of biological material. This is not an invasive procedure, just blood taken from the finger. Here I did not translate this timetable because I think all these metabolites are well known, just to remind the colleagues that all these changes on the results of tandem spectrum are not some unknown indexes. Each combination of various metabolism, each combination speaks about some specific disease. When we see a high level of all materials, then probably there was some problem in the way the sample was taken, or it was dirty or badly transported. We consider the combination, and each metabolite can speak about this or that diagnosis. As for the results of TMS, this is research which was held in the US, and it reminds that each and every metabolite we can see as a result of this procedure has its own sensitivity and specificity. These are serious requirements for the test which we use to base on the results to make the diagnosis. Some of them are sensitive to nutrition or to how the skin was cleaned, or some of them make almost no mistakes, and their changes are considered important to suppose that there is some hereditary problem. What should you do if you have a positive analysis? In fact, there are two ways which are divided by which are different by the consequence of activities. First, before the positive diagnosis, as a rule, this is DNA diagnosis, and after having this diagnosis, their special therapy begins, or not to begin with, if the diagnosis is negative. The second way is when we begin treatment even before we have the proof of diagnosis. Unfortunately, with hereditary cases, we select the second path, BOA, more often, because we begin with a therapy waiting for the results which prove the diagnosis. In many cases, if it's not there, the results proving the diagnosis will be when it is too late for the patient. What tests of the second line do we have after we got some changes as a result of a blood analysis? Organic organic acids in urine analysis. Here you can see the level of metamolon acid higher, over 2. As a rule, it's not 4 or 5, it's over 1,000. These are the numbers which speak about metamolonsiduria. After positive results, which we use after this analysis, we can quite positively start therapy, waiting for DNA diagnosis in parallel. And the next diagnosis, there is a number of options. If we are sure about the diagnosis, and there is a faster and simpler analysis when we make analysis from one gene on often mutation. Unfortunately, it is not often applicable in cases if there is a marriage between the relatives, so the patients are of a different ethnical origin. We cannot judge about the mutations. Then we're speaking about more complex and long analysis, and that's the full analysis of gene to find more rare mutations. The next phase, we can send it to target panel of genes. I will tell about that on the example of panels which we use very often. And the next one is full exome or genome sequencing. All these analyses can be held today with dry blood if they were taken properly, if they were not for long in the open air, so taking DNA from the filter is possible today. As for target gene panel, what is that? We closely cooperate with the gene laboratory of the center, specialized center, where there are specialized target panels. And here, as an example, I can give a panel which is called hepatopathies and hypoglycemia. The patients who have this diagnosis sent there, and the doctors cannot select a gene with a probability of 100%. And here is the result of 168 patients. In 80 cases, the diagnosis was confirmed. That's a high percentage for such panel, and you can see that most of the patients were diagnosed with glycogenosis, most of them, and different types were there. Alagille syndrome and other rare pathologies. Getting back to our three stories, three girls, 3 months old. The first was delivered with complaints of delay in failure to thrive, increase of abdomen size, bleeding from the places of injections, anamnesis preterm born. At the same time, it was discharged from the hospital on time. From 1 month old, the parents found out the increase of the abdomen size. What we have to say as well is hereditary anamnesis. The older sister died when she was 3 years old with cytomegalovirus hepatitis, and it was proven with antibodies and other diagnosis. That time it was the leading diagnosis. As for the examination, there was an increase of the size of her liver. Baby was unconscious without focal neurological symptoms. As you can see on the screen, the analysis of biochemical tests. And coagulogram showed quite significant coagulopathy. So there was MS results, increased tyrosine level, increase of succinylacetone level. This is curable today, and now there is a genetic therapy cured by nitisinone, parallel to low protein diet. This is a picture of this girl of the New Year celebration 2020. She was fine, she was singing songs, and she was all right. If the therapy starts on time, the forecast is more or less positive. Of course, the child is still being observed because there are some threats of cancer, certain level of threats, and it's harder for her to be low in proteins. The second case, girls 3 months old with complaints of fever, refused to eat, once generalized seizures and sleepiness, anamnesis. It was full-term born, discharged from the hospital on time, and up to 3 months showed no complaints. She was rapidly brought to hospital without any predecessing problems. Increase of liver up past 3 centimeters. Sleepy child, did not shout, did not react. Metabolic acidosis, and the analysis you can see according to the slide. And unfortunately, this is not introduced in routine practice of ambulance. The measure of KFK. This index helped to find the correct diagnosis. It was the CK level was 11,000 per liter. No problems with coagulogram. This composition of high level of CK and reduction in GLU and absence of any anamnesis and presence of pathology can lead to an idea of hereditary problems, and as a result of the survey, diagnosis was fatty acid oxidation disorder, altered mitochondrial morphology, and it is curable. Treatment is low-fat diet, frequent nutrition, and MCT oils additives. Unfortunately, such children are observed with diagnosis of reactive hepatitis of unknown etiology, because there is a fever, there is temperature, and they vomit, and biochemistry changes are referred to that. Unfortunately, without proper therapy, these children die rapidly, and diagnosis very often comes when it's too late. And the last case, a girl of 3 months old, all the complaints the same, more or less the same, very similar. Anamnesis full-term born, and she is going to hypoglycemia, and the child was taken to ambulance. They saw increase in the size of abdomen, and there was acid liquid in the abdomen. As for the hereditary anamnesis, older sister died when she was 7 months old. The diagnosis was not there at the moment of death. And the reason of death was digestional bleeding, gastrointestinal bleeding. The liver was increased, ascites, and the child was not only unconscious, it was in good spirit. She was smiling, reacting, no focal neurological symptoms. Metabolic acidosis with lactate level increase up to 10, BE minus 18, GLU 1.8 mmol per liter, biochemical blood tests, liver ferments are increased, and reduction of albumin for 18 grams per liter. At the same time, fibrinogen was not detected. It was so low that it was impossible to detect. At the same time, hemorrhagical syndrome was not there from the points of injections, even no blue marks on the skin. It was considered as some problems with the liver, and as for the following picture, biochemical one, and it was done for quite long, and it helped a lot. Hereditary anamnesis. The oldest sister was examined seriously before she died. That's why many diseases could be excluded right away from diagnosis search. As for the results of MS results, increase of tyrosine level, increase of alanine level in this situation. It's not really specific. These are metabolites which very often can be seen in any damage of the liver. Tyrosine can go up in alanine in many mitochondrial diseases and in high polyorganic absence or insufficiency. This picture on MSMS did not show us any how to find out the diagnosis, because it was obvious that the child has problems with liver without this analysis. As for the urine analysis, there were high lactates and ketosis signs, narrowed the spectrum to mitochondrial pathology. There were 2 mutations discovered in TRMU gene. This gene is a complex one, an interesting gene, which is described and is linked to the development of liver failure, and patients which manage to live through this liver failure, they manage to live and grow without any significant problems, and request some more attention to than a patient with mitochondrial pathology. But most of patients described with this diagnosis died when they were there. That's why with approach to therapy, it was really hard to find that we sent this baby to transplantologists for liver transplantation. I would like to remind that it's a very simple analysis. It's not an invasive one, and making this diagnosis is important both for the patient and for the doctor. It was clear even earlier from the cases. It's important for the family, for planning healthy pregnancy, and for us to understand how to treat patient, and for our own experience in order to give diagnosis in a better way. Thank you for your attention. If you have any questions, I'm ready to answer.
Thank you very much, Natalia, for very interesting information. Dear colleagues, are there questions? Do you have questions? My name is Maxim, and my question is as follows. Very often the couple is not ready, not prepared to become parents. It's very difficult to get ready. They have no experience how to observe a child, what they should pay attention to. And very often, since they can go to private hospitals, to public hospitals, it's my experience at least, all the doctors spend just a little time with patients, and they do not have personalized conversations in order to reveal the complaints and some pathologies. Are there any requirements or methods of genetic diseases diagnosis based on the data of analysis of tests? Are there any systems which allow to reveal such diseases? And question number 2, how the doctor should behave, because the parents are afraid to be called bad parents, that they do not pay much attention to the child. How a doctor should talk to parents, and how to make them tell all the information that they are hiding, probably they would keep secretly, because they just don't want to seem bad parents. They come to different doctors, tell one doctor one thing, another thing to other doctor, and they are trying to make a diagnosis themselves. Could you please tell me if you're a doctor or you're a patient? No, I'm not a patient, I'm not a doctor. I'm just a curious person. We don't have much time, that is why. Please answer the question, please. remember that we are almost out of time. My first question was about some automated system of diagnosis when you enter data and the system gives you an answer. Not only system, but any methods, techniques. Well, actually, methods. Screening is something that I was telling about. It's a method which allows us to make our search narrow, in a more narrow area. There are internet resources. I don't mean Google. You should read Google your disease. You don't. There are specific programs, genetic programs, where we can find some hereditary diseases with symptoms. We can just read about some symptoms or something that will make us suspicious and will make a patient go to a doctor. For example, we are offered a list of diagnoses where we can choose the one which fits. How to talk with parents if they do not reveal information. I believe there are some general recommendations. I think that parents from the very beginning, if it seems to you or a doctor thinks that parents are not quite open, you should remind that it's their responsibility and it's our responsibility. We came to a doctor, they are also responsible for everything they say to a doctor, and we can make our conclusions only on the basis that they have, and also the government of Moscow took care of that, and during some days there is an access to the unified medical documentation of every patient, and if the parents go to the municipal hospitals, but not private, unfortunately, yes, only public municipal hospitals are included in this system. My vision is that of this problem is that if we establish a system which would be anonymous, because if it's anonymous, the parents will not be afraid of telling everything. It will allow a doctor to popularize resources, to promote some resources where they can get more information, not only when they go to doctor. Well, to be honest with you, there are such resources in Moscow, and there are very strong patient organizations, associations, social associations which support parents. I cannot name them at the moment. The question about popularization, information resources, and probably a doctor whom you visit, whom you trust, will give you advice. Is it a requirement? Does a medical personnel has to inform about the resources where we can find more information? Because my child is 2 years old, and thanks God everything's fine with my child, but no doctor ever told me about the resources where we can find information. No, there is not such requirements. It's your curiosity. Your child is fine, and I mean resources, then you suspect that you have any disease. If it's not prevention, it's a method of curing. Prevention is a wholesome lifestyle and everything we were discussing. Thank you. Dear colleagues, we have more questions. I'm Elena from Samara. I have a question to Natalia. Could you tell more about TMS test? Is it available in the regions, not only in Moscow, and how can we take part in that? And this test is for acids. Isn't it the analog of other text, urine test for amino acids? Because it's also acids. This method is different. The regions, of course, can have this test. In federal tests, laboratories, federal centers, laboratories, yes, you're right, that you can send dry spots, dry blood spots, usual postal. What is the price? Is it the same price that amino acids? the price is about 2,000 rubles. Thank you very much. More questions? Dear colleagues, I believe that we can discuss it, everything we want after. Unfortunately, we have not covered all the topics we announced, but I believe that our program is very busy. We did not expect such a deep interest. If we have expected that, we would probably reduce the time for presentation and increase time for discussion. The topic is very specific, and I believe that now you have an opportunity to talk to us. We are here on the platform. We will answer all your questions, and I would like to thank the panelists, the participants, and I would like to say that the patients with a hereditary disease, I liked the question about spirit and the will. I would like to conclude with the words by Goethe: only one deserves life and freedom who fights for it every day. And families with a child with hereditary diseases lives every day with that. They fight every day. And Moscow Healthcare Department, today we are here with best practitioners. International community will invent all the efforts in order to facilitate and to make life of these people easier and to cure our children. Thank you very much.